Hello and welcome to this webinar, COVID Targets in the Crosshairs, Unparalleled Quality and Speed from Arctoris' Ulysses platform. I'm Lou Rahman, Editor-in-Chief at DDW. Today's webinar will be presented by Catherine Walk, BMG LabTech, and Dr. Daniel Thomas, Head of Discovery Biology at Arctoris. COVID-19 has highlighted the current operational vulnerabilities in pharmaceutical research and in the drug discovery pipeline. Bringing new drugs to the market is still a slow and expensive process with far too high costs, failure rates and cycle times. With low reproducibility and inefficient processes delaying the path to approval, the pandemic has prompted a fundamental reassessment of strategic priorities. Dr. Daniel Thomas, Head of Discovery Biology at Arctoris, is a drug discovery professional with over 20 years of experience in the pharma and biotech industries. In this webinar, he will describe how Arctoris' automated platform, Ulysses, helped to accelerate onboarding and validation of 3 critical assays targeted on Sarskov2 drug discovery. Case studies will focus on key target functions such as viral processing, infectivity, and transmission. The webinar will also discuss how automated experimental and analytical workflows can deliver superior quality, data, and expedite project timelines. It will include 3 COVID-19 case study examples to highlight the attainable data quality and will explain how microplate readers support robust assay performance. Daniel has extensive theoretical and practical knowledge of assay development, molecular profiling, and enzyme kinetics. He is an accomplished leader with a track record of developing transnational matrix research teams and leading early-stage programs across a broad range of therapeutic areas. Catherine Walk is the Applications Manager for BMG LabTech UK. Her team is responsible for the support of customers in the UK and Ireland. She has 16 years experience with BMG LabTech, assisting users with microplate reader and assay optimization, drawing from her previous experiences in both academia and industry working in the fields of cell and molecular biology. The presentations will be followed by questions. We will begin with Catherine, who will be looking at innovative microplate readers designed for drug discovery. Catherine, over to you.
Thank you. As sponsor of today's session, I'm going to be taking a few minutes ahead of Daniel's talk to both introduce BMG LabTech to those of you perhaps not so familiar with us as a company, and also to introduce 2 of our microplate readers, the Clario Plus and the Ferrostar FSX, as these detection instruments play a key role within the Ulysses platform at Arctoris that we're going to hear more about today. BMG LabTech is a German company with more than 30 years experience in the production of high-performing microplate reader instruments. We offer single-function and multi-mode readers able to accomplish both basic research through to drug discovery and high-throughput screening. So, to introducing our microplate readers, the Clario Plus and the Ferrostar FSX. Both of these instruments are modular and upgradable and are capable of reading to the highest sensitivity levels. Both can read all non-isotopic read technologies as shown below. Both of these instruments are somewhat complementary and therefore share some common features, such as the ultra-fast spectrophotometer that they use for absorbance. This allows users to scan between 200 and 1000 nanometers in under 1 second per well. This speed cannot be matched by other monochromator type devices. It can also be very useful when looking at things like compound interference or we're just looking to optimize absorbance assays. When using glow luminescence, crosstalk can often be an issue where you get light from above and across the microplate interfering with the reading, in addition to light coming through the microplate. We offer advanced crosstalk reduction to eliminate these effects. We also have top and bottom reading using Z focus. This allows us to optimize for different volumes in the well and allows us to achieve the best performance when reading from monolayer in bottom read mode. In terms of standard features, we also offer temperature control and user-defined shaking to optimize assays. When measuring cell-based assays or non-homogeneous sample types, both instruments can use well scanning options. These approaches are suitable for all plate formats up to 384 well microplates and can improve data quality by reducing the variation between replicates across the plate. Our averaging approaches, orbital averaging and spiral well averaging, allow us to rapidly measure multiple points across the well, returning one value representing the well. When wanting to get a better idea as to the distribution of signal across a well surface, we can use a low resolution imaging type approach called matrix scanning. This allows users to measure up to 900 points per well and to be able to work with, say, thresholding within the well in addition to getting average values for the well. So it's a great way to understand what's happening with your population of cells. The rapid nature of these approaches and the small data files produced make this approach very suitable for even screening cell-based assays. So far I've spoken more to the similarities of these instruments, and I'd like now to talk a bit more specifically about some of the unique features of these tools. When I think of the Clario Star Plus, I often think of it with respect to assay development due to its flexibility in addition to its performance. It includes a linear variable filter monochromator that allows us to scan both fluorescence and luminescence, which can be very important when doing assay development prior to a screen. As mentioned, it includes a spectrometer but also an alpha screen laser, and it's able to read all plate formats up to 1536 well plates. With injectors for a reagent addition, it's capable of measuring kinetic style measurements, and it's ideal for cell-based assays as it also has, in addition to temperature control, the ability to independently control CO2 and oxygen to create physiological conditions. The patented Lvf Monochromator is unlike any other monochromator design in that it offers filter-like performance. It allows users not only to dial in the wavelength of interest, but also to adjust the bandpass, the amount of light that's measured at that wavelength as well. This offers a real unique flexibility and allows users to reproduce settings in journals or past methods. From the drug discovery application side of things, I've listed here a number of drug discovery methodologies. We can measure everything from endpoint through to even fast kinetic responses, and the Clario Star Plus is really an ideal tool for any low to medium throughput laboratory. When thinking of higher throughput requirements, many would consider the Ferrostar FSX. It was designed with sensitivity and speed in mind. For many HTS labs, it would be considered a gold standard, with its ability to read up to 3, 4, 5, 6 well plates. Its dedicated read technologies such as the spectrometer, alpha screen laser, photon counting detectors and laser for TR-FRET make it the industry's leading instrument in its class. It offers simultaneous dual emission in all readouts, including alpha screen, and its inject and read capability at the point of measurement means that even the fastest kinetics can be captured with this instrument, which is particularly useful if looking at on and off rates, for example. From a drug discovery assay perspective, the Ferrostar FSX can accomplish all those applications shown for the Clario Star Plus to the highest of sensitivity levels and speed. The simultaneous dual emission in particular is a benefit for the FRET, BRET, FP or TR-FRET type applications where they have a dual readout, and including alphaplex as well, where wanting to measure multiple pathways in the same well. With respect to automation, both instruments incorporate barcode reading and are stacker compatible. Our stacker magazines can hold up to 50 microplates in this continuous feed design. But if not using a stacker, users can also incorporate our instruments into robotic automation platforms, and support is available to assist anybody in doing this, as BMG show no preference to any robotic provider. When wanting to optimize LIMS data handling, support is also available from the BMG applications team to assist in doing so. If not already using a data analysis package, or when doing assay optimization on a smaller number of microplates, users may wish to adopt our MARS data analysis software. MARS stands for Microplate Analysis Reduction Software. It's provided with each of our instruments and is a license-free multi-user platform. Utilising MATLAB type algorithms, we're able to provide automatic data reduction with users able to create specific protocols and apply templates to data. As mentioned previously, it's very easy to get raw data out of the system automatically, especially for use with LIMS system, but just as easy to get analysed data from our MARS software. We can do an extensive range of applications, including quite complex kinetic analysis, whether it be standard curves or IC50s, statistical analysis for standard and robust statistics, and also assay quality parameters such as Z prime, signal to blank and percentage calculations, with full reporting available in a number of formats from Excel through to PDF and HTML. To summarise what these instruments offer, through their high performance optics and design, they offer the highest data quality. We're able to provide useful data analysis tools and easy export and compatibility with LIMS systems, and from an automation perspective, through recording through barcodes, stackers, and the ability to work with all robotics platforms, our systems remain easy to automate. Well, I will finish there and say thank you, and it's now my great pleasure to pass over to Daniel for our main talk of today.
Thank you, Catherine. We'll now hand over to Daniel, who will explain how Arctoris' automated platform, Ulysses, helped to accelerate onboarding and validation of 3 critical assays targeted on SARS-CoV-2 drug discovery.
Thank you very much for the opportunity to speak at this webinar and to introduce how Arctoris is collaborating with BMG LabTech to drive development of its fully automated Ulysses platform. Arctoris was founded in Oxford just over 5 years ago and earlier this year celebrated its 5th birthday. Whilst our journey is only just beginning, we are focused on alleviating the crippling issues of irreproducibility and attrition and the spiralling costs that challenge the very foundations of research. How? By going back to basics, eliminating variability at its source by digitizing experimental and analytical workflows, coding scientific theory and manual processes to guarantee consistency time after time. We do this using precision automation, established technologies, and working with data scientists and software engineers to deliver a comprehensive, personalized data experience. During the course of this webinar, I shall highlight the fundamental challenges facing drug discovery and how the current pandemic is directly influencing change. I will introduce Arctoris' philosophy and how we are creating a unique value proposition. The critical importance of data quality, including sources of error and variability, a reminder of the differences between precision and accuracy, and how innovation is as much about looking forward as it is about understanding where we have come from. I will then introduce Ulysses and describe our automated experimental and analytical pipelines before walking you through 3 COVID case studies. But first, I'd like to reflect on the fundamental challenges facing drug discovery. It is encapsulated in a thought I jotted down in 2013 and have shared in many presentations since. Writing this webinar gave me time to reflect on how the pandemic has brought the challenge into even sharper focus, leading me to add an additional perspective directly influenced by Davis Smith of Cotopaxi for today's audience. The growing pressure to deliver greater value with fewer resources is a familiar concept that has been challenging the drug discovery paradigm for more than a decade. The current pandemic has highlighted operational vulnerabilities which must be addressed, but has also given us an unprecedented opportunity to act, to leverage partnerships, serve the greater good, go virtual, and create a shared purpose. Comparing the photos, it is evident that research labs haven't fundamentally changed since the 1960s, and despite improvements in technology, such as high-throughput screening, biotechnology, combinatorial chemistry, and computational drug design, drug discovery is becoming slower and more expensive over time, a trend first observed in the 1980s and named Irum's law. Poorly annotated and unstructured data and inefficient use of valuable resources result in some shocking statistics captured in a number of reviews over the last decade. Almost 90% of landmark oncology data published cannot be reproduced by independent groups, and as many as 50% of cell lines used in labs are either contaminated or misidentified. Moreover, with hundreds if not thousands of new publications every day, we are struggling to keep abreast of the latest literature. We are spending a disproportionate amount of time on repetitive manual tasks which could be more efficiently and effectively executed by automation. Embracing a future that protects our most valuable resource, our research scientists, through collaborative engagement with expert-led CROs will ultimately usher in a more productive and successful future, one where we spend more time reading the literature, designing higher value experiments and interpreting the results, and ultimately making better informed decisions. Arctoris operates a centralized, fully automated drug discovery platform, enabling scientists to conduct R&D remotely with precision, accuracy, transparency, and full reproducibility. We are not a high-throughput screening lab. Instead, we enable end-to-end automation of entire discovery functions, leading to advanced data quality and full metadata capture across the whole discovery path. Our processes guarantee experimental consistency, capture more data per assay, reduce cycle times, and increase convenience, enabling automation of low-throughput work as well. All this while completing experimental onboarding faster and providing secure and convenient data storage to clients. Successful drug discovery requires high-value, reliable data. Therefore, data depth and full data capture are critical. Currently, we are heavily reliant on unlinked, isolated datasets, frequently generated using ambiguous methods, collated in non-standard formats, and stored in fragmented storage systems. This makes the data extremely difficult, if not impossible, to access, and reuse is out of the question. We are hemorrhaging value right from the word go. At Arctoris, we have built systems that ensure data is generated using verified reagents and standardized protocols. It is captured and digitized together with a fully transparent, comprehensive audit log and stored in an interoperable database. These processes ensure experiments can be repeated exactly, building confidence in the data generated and enabling better decisions to be made faster. Accumulated variability is the real Achilles heel affecting reproducibility and is often overlooked as being not important. However, if we add all the seemingly negligible variables together, we can end up introducing significant variation. Variability and errors come in all shapes and sizes, from the measurement errors of validity, reliability, and repeatability, where we are assessing the ability to accurately measure and remeasure an effect, through to sample and observer-based variation. Here, different samples can produce different results due to chance, resulting in false positives or false negatives, although natural variability in reagent will also impact outcomes. The observer can also introduce significant variability, inconsistencies resulting from a single observer recording repeat results, through to a failure of different observers to record the same result. Systematic errors, technical limitations, and system faults frequently, but not always, arise due to poor training and maintenance practices. And finally, random or chance errors are just that. More often than not, one cannot predict them, but they can be mitigated through implementation of standard processes and a control framework. The impact of error accumulation is not always obvious, so I created this simple visualization, which I've named the witch's broom of variability, to highlight how quickly errors can lead to divergence from the true value. Exploring the influence that errors ranging from +10 to -10% can have on the final outcome when introducing just 3 subjective intervention points is quite enlightening. What is certain, however, is that the accumulation of errors will compromise discriminatory power, affect data accuracy and quality of decisions, leading to increased costs and cycle times. Following the cost of quality rule, it costs $1 to verify a record as it is entered, $10 to correct it, and $100 if nothing is done. It is critical, therefore, to get it right first time. The journey from hit to candidate is fraught with challenges, least of all the complexity of the biology that underpins disease. Our ability to navigate has dramatically improved over the last few hundred years, starting with pure luck and chance, using the position of the sun, moon, and stars, then radio positioning and radar, and now GPS. We have exploited advances in technology and automation to enable us to know more precisely where we are at any given time, and it is these same principles that we are applying to early drug discovery processes to deliver better data and accelerate improved decision-making. We have all seen these dartboards before, representing precision and accuracy. From our perspective at Arctoris, precision is how close measured values are to each other. It is defined largely by the process and requires the elimination of variability. Accuracy, however, is how close a measured value is to the true value. It is largely defined by the biology and requires functionally relevant assays and a predictive cascade to ensure translation from simple to more complex systems. Innovation continues to deliver amazing advances in research, from the elucidation of the structure of DNA to CRISPR gene editing, the discovery of penicillin to the design of the first gene therapy, and moving from single sample cuvettes to 1536 well plates, enabling the rapid profiling of millions of molecules. The quality of a decision is directly related to the quality of data used to inform it. The best decisions come from the best data. These are typically higher content, richer and broader, and gathered in parallel, rather than single pieces of evidence gathered sequentially. Some of the most important technologies in use today have been around for decades. Think robotic arms. General Motors introduced the first industrial robotic arm called Unimate for spot welding in 1961. Think inkjet printing. Hewlett-Packard's ThinkJet printer launched in 1984. It uses a pulse of current through a heating element to vaporize ink in a chamber, forming a bubble which causes a large pressure increase, propelling a droplet out. Think positive displacement pipettes. Gilson's Microman, where the liquid is in direct contact with a disposable capillary tube and piston, allowing the aspiration force to remain constant, enables the pipetting of a huge variety of different liquids. All three of these technologies are reliable, trusted, and proven to work, and now underpin key processes on our platform, having all been reimagined for use in 21st century laboratories. At Arctoris, we are building fully automated workflows, integrating experimental and analytical capabilities to deliver the highest quality datasets to researchers around the globe. Our automated experimental workflows have been established using key platforms built on trusted technologies, as I articulated before, including independent non-contact dispense capabilities aligned with state-of-the-art multimodal plate readers. BMG plate readers deliver the speed and quality of data acquisition, enabling real-time monitoring, supporting detailed kinetic assessments, and ensuring mechanistic planning is incorporated into projects as standard from the outset. The exquisite resolution eliminates serial dilution and facilitates unmatched experimental design potential. Direct, independent non-contact dispensing minimizes reagent usage, reducing our carbon footprint and costs. Comprehensive protocol encoding ensures unparalleled reproducibility, and seamless integration delivers reduced cycle times without compromising quality. Our automated analytical workflows use encoded scientific theory and statistical models underpinned by a number of proprietary algorithms to deliver unique data-driven potential. We have developed pseudo-concentration response curve methods to estimate potency, enabling targeted full curve dose response design, reducing the potential impact of false negatives by highlighting data trends at or below the robust cutoff. Pseudo-CRCs coupled with targeted full curve dose responses can also be used to more precisely refine the ranking of closely related potencies. Indicative mechanistic profiling provides contextual information, augmenting outputs, and it is this enhanced mechanistic surveillance that can assist in the early identification of opportunities for differentiation. Finally, comprehensive mechanistic data packages can be generated to support key milestone decisions, such as molecule acquisition, candidate selection, and IND filing. Custom pipelines enable the development of personalized proposals tailored to the research project. Innovative development of traditional experiments focuses data to the regions that add the most value. Digitization of theory facilitates experimental design at a molecular level, enhancing dataset value, reducing the need for excessive replication. And unparalleled reproducibility enables differentiation of more closely related potencies. In order to effectively digitize theory in high definition requires exquisite flexibility and resolution, both temporal and volumetric. If we compare traditional approaches for some key experiments with those developed at Arctoris, we can start to see how the independent, targeted delivery of reagents enables more data to be generated in the regions that matter, enhancing the value of the outcome. With single-shot or similar approaches, we are developing pseudo-concentration response curve methods to enhance the value of data generated in focused hit ID campaigns. In traditional single-dimension full-curve profiling, front-loading the experiments with a pseudo-CRC pre-screen has enabled personalized profiling to be offered, providing individualized responses for each molecule, enabling resolution of much more closely related potencies. In later stage multidimensional characterization, we are able to deploy independent dosing to print theory, enhancing the ability of statistical tests to discriminate between different mechanistic models. The enhanced definition we can achieve with this resolution will really help drive better decision making. The Sarskov2 pandemic marks the third zoonotic introduction of a highly pathogenic coronavirus into the human population. Recent SARS-CoV-1 and MERS-CoV epidemics raised awareness of the need for clinically available therapeutic or preventative interventions. However, the development of effective intervention strategies relies on the knowledge of molecular and cellular mechanisms of coronavirus infection, which highlights the significance of studying virus-host interactions at the molecular level to identify targets for antiviral intervention and to elucidate critical viral and host determinants that are decisive for the development of severe disease. In this schematic showing the key biology, I've highlighted intervention points that we have rapidly built and made available assays to monitor inhibition of key targets. SARS-CoV-2 infects human respiratory epithelial cells through direct interaction between spike and a number of receptors, including ACE2. It is these interactions, essential for binding the virus to the host cell at the start of the infection, that play a critical role in host specificity and viral infectivity. The spike RBD-ACE2 interaction is a key target for vaccine design, with neutralizing antibodies blocking viral infection by binding the viral spike protein, preventing interaction with ACE2 via mechanism 1, or binding the viral spike protein, blocking conformational changes required to facilitate fusion of the viral host and host cell membranes via mechanism 2. We have also made available FRET assays against 2 protease targets involved in viral polyprotein processing, and are exploring others responsible for supporting viral RNA synthesis. Case Study 1 focuses on targets involved in the human immunological response to COVID, rather than those viral targets directly responsible for infectivity. Alongside a fully annotated process timeline, it showcases full potency, selectivity, kinetic and mechanistic profiling of a set of 16 late-stage lead molecules for a biotech partner. We used the Janus family of kinases to demonstrate the speed with which we could onboard assays, achieving reagent to pharmacological validation of 4 isoforms in less than 100 hours. In collaboration with our biotech partner, we couriered molecules from chemistry CRO to Arctoris, monitoring temperature throughout transit, and processed these on our Ulysses platform, where all assays returned robust Z primes in excess of 0.8. We delivered kinetic and selectivity profiling, together with indicative mechanistic analysis, all in less than 48 hours. Data revealed kinetically differentiated mechanisms, enabling new molecular stratification, with our biotech partner planning a new SAR MOA cascade to drive lead optimization with sub-24-hour data turnaround. Case Study 2 highlights the accelerated onboarding of one of 2 sentinel COVID proteases, namely the 3C-like main protease, completing comprehensive reagent characterization, assay encoding, and platform validation in under 10 hours with robust Zs in excess of 0.9. From the initial enzyme titration, iterative substrate affinity assessment, and follow-up enzyme and product titrations, through to assay robustness assessment is shown on this process timeline. Full process maps, experimental blueprints, and analytical pipelines were constructed and validated in less than 7 days for both the 3CL and papain-like proteases, ensuring rapid access to key viral target assays with molecules to data possible in as little as 3 hours. Finally, Case Study 3 demonstrates the value of collaborative engagements, which here enabled early access to a key viral assay which monitors the interaction between human ACE2 and the viral spike protein. The availability of high-quality reagents and assays facilitated onboarding and provided independent validation, guaranteeing rapid data turnaround. Use of state-of-the-art dispense and read capabilities enabled autonomous reagent addition, minimized dead volume, and ensured exemplary levels of assay performance. We achieved successful onboarding within 1 day of receipt of 2 200 data point 96-well pre-release kits. This included delivery of a robust, reproducible assay with signal to background in excess of 20:1 and Z prime in excess of 0.8, alongside confirmed displacement using untagged recombinant ACE2 and spike control proteins. Final validation was confirmed by near identical pharmacology observed for positive and negative control neutralizing antibodies. At Arctoris, our journey may only just be starting, but already our concept is being adopted by groups across the full scientific spectrum, from academic groups to virtual biotechs and large pharma organizations. Our automation solution delivers a single, centralized platform offering a validated library of drug discovery protocols, deep mechanistic profiling, and a broad range of cellular R&D capabilities across early drug discovery, providing a fast, cost-effective, reliable and accessible platform to researchers across the globe. Thank you very much for your time, and I'd be happy to take any questions.
Thank you, Daniel, and thank you both. They were great presentations. Before we continue to questions, I'd just like to remind everyone that this webinar will also be available to access on demand on the DDW website, www.ddw-online.com. We're now just going to pause for a few moments to allow the audience to send through any questions that they might have for the presenters. Okay, if you're ready, Daniel and Catherine, we'll go to the questions. So the first one that we have is what cycle times are you achieving in reality, and how quick can you really turn around from receipt of reagent stroke compound to meaningful data?
Yeah, thank you very much. So I think largely it depends on the nature of the assay at hand. Clearly, assays that have poorer activities, or where the protein reagents or cellular reagents are unknown or poorer quality, it can take longer. And if we're talking assay development, clearly that is a much longer process altogether. So really what we are referring to by onboarding is those final stages of assay parameter refinement. And typically, this can be done in a biochemical setting in under a day. Our record is, I think, about 6 hours. In terms of cell-based arena, that's clearly going to be dependent on the cell line, what your endpoint is, and clearly that can be, that could be a week, that could be a month. It really depends on the assay at hand, but it can be done significantly faster through coding of sort of sentinel experiments in there.
Okay, thank you. And the next question, this is a really good question. Do you see the research landscape changing in response to COVID-19, and if so, in what way?
Short answer, yes. I mean, I think it's forced a lot of researchers, a lot of companies to take a look at the contingencies they have in place for responding to issues of global significance, freedom of movement, the ability to maintain continuity in processes when things like pandemics prevent staff getting to work. So I think it has triggered a fundamental reassessment, and we've certainly seen that in real time, with companies coming to us to ask, what we can do to assist. So very much so. I think it's going to continue moving in this direction.
Yeah, I would add to that as well, that I think having agility has been one of the keys to succeeding under the circumstances that we've been working with, and people looking to companies like Arctoris to support their the activities that they're working on. It becomes ever more important, I think, for the future. And I think that there's been so much good collaboration happening as well at this time. And you agree, Daniel, that it's been wonderful to see.
Yeah, I suppose it would be a real shame to go back to the I call them the bad old days of too much competition, holding things back. Collaboration really has seen us get from not knowing about the pandemic 18 months ago to having a vaccine. So it really demonstrates that if there's a will, there's a way. Now, yes, it costs money. Yes, economies demand that happens. And I'm not suggesting that small molecule drug discovery will necessarily accelerate to the same degree, but it should certainly not take the 12 to 15 years that it does to date, and it shouldn't fail at the level that it has done for the last, more than the last 20 years. So, collaboration will be the way that we see better advances, better medicines, and hopefully, higher quality outcomes.
Okay, thank you. They're great answers from both of you there. I think this next one's for you, Catherine. It's both the Clario Plus and the Ferrostar FSX are used in the pipeline. How do these instruments compare, and are there advantages of one over the other?
Okay, my internet was breaking up a little bit there, but I think I heard you say about what would the how do they compare, maybe. So the Clario Plus is a monochromator based instrument that allows you to define wavelengths very easily. So often it's considered as an assay development tool when starting out with assays, working out concentrations and those sorts of stages. The Ferrostar FSX offers similar, the best sensitivity that we can provide and the fastest read times, in particular for FRET type applications. So for fluorescence and luminescence, we may see similar performance on the Clario to the Ferrostar, but maybe speed advantages, especially when there are dual emission readouts. And for things like fluorescence polarization, HRF, for example, and HRF is probably one of the biggest drivers towards the Ferrostar FSX. In its that it uses dedicated detectors for that read technology and allows things like injection with instant read, for example. So you know, the two instruments work really well as a combination within a lab that has to deal with different stages of the drug discovery process. So they complement each other really well, and they each have their strengths, if you like, in terms of, you know, assay dev and being able to trial different wavelengths on the Clario. And then when you're in that screening modality or wanting to work with very rapid kinetic assays, for example, especially with dual readouts, that's where the Ferrostar really comes into its own then as well. I mean, in your experience, Dan, is there, you know, how do you see them fitting within the line at Arctoris?
Yeah, I mean, I've been a convert for many years, but I think the advantages and the advances that have been made over the last probably decade or so have really seen both sensitivity increases, you know, dynamic ranges, the flexibility. I mean, back when I was an academic, my real bias was against multimodal readers, because they kind of fitted, they were jacks of all trades rather than masters of a single one. But actually, what you guys have been able to land on is something that actually fits the bill that we need, which is, you know, a single go-to technology covering a reasonably broad range of modalities that allows us, you know, flexibility, sensitivity, speed, and for us, you know, speed and quality, as I articulated in my talk, are absolutely central to being able to drive the best decisions in a timely fashion. So I would echo your thoughts that we see the Clario really acting as the workhorse of ensuring that we can get the assays into the system, and then once the coding, you know, takes over and integration with Ferrostars gives us that real speed to deploy, you know, the work cell to deliver, you know, what we trust will be delivered from the outcomes that we've got out of the Clario. So they complement each other very well, and the flexibility, you know, enables you to, you know, scope a broad range of areas before locking them down with the sensitivity and speed and precision that the Ferrostar brings. So yeah, it's
great. Yeah, thanks, Dan.
Okay, thanks both for those responses. The next question that we have is, in terms of automation solutions that you've presented, why did you choose to use what you have when there are so many on the market?
Again, I'm a bit of a creature of habit, but I do defer. I think I mentioned in one of the slides, to technologies that are tried and trusted. That's not to say that innovation and new technologies don't have a place. But if we are wanting to run, you know, an autonomous work cell with the confidence that it will not fall over, not break, you know, what we rely on is real precision engineering. And in inkjet printers, in positive displacement pipettes, and in robotic arms, we have all three. And largely, moving fluids around is the one thing that a robot can do, you know, time infinitum. You know, it can keep doing it the same way every time, whereas a human will clearly, you know, get tired and make mistakes. So, you know, what we're looking for is alleviating the manual elements and trusting technologies that have been around and have done this in other industries as well as our own for years. So, you know, I've always been one for collaborative engagements, you know, working with vendors, working with innovative and imaginative collaborators to identify how, you know, a technology could be repackaged, or, you know, where there's a gap, you know, can we work with a company to fill that? You know, it's not a one size fits all, but at the same time, you know, as the question highlights, there are plenty of solutions out there, and we are always, you know, scoping, you know, new technologies. We we need to have confidence that they will perform, but there's always opportunity to bring new technologies in as well.
Okay, thanks for that, Dan. So Catherine, the next question is, what LIMS systems are compatible with BMG LabTech instruments?
Okay, so from a LIMS perspective, it's always possible to recover raw data from either the Ferrostar, the Clario, any of our systems in an automated way. So that data can be sent on either a protocol specific basis or on a user account basis to a location of the user's choice. This data can also be presented in a number of different formats, in either table views or microplate views, so lists or microplate formats, and that can then be picked up by the LIMS system of choice. So usually to work with the LIMS, it is really based on a requirement to get either text data, CSV, or Excel-based data that LIMS system can pick up automatically. So we have a lot of scope to create those formats and work with all leading data management systems, so we can really assist users with whatever format they'd like to work with. So no issue at all with any form of LIMS system. We can work with everyone.
Okay, thank you for that. And the next question that we have is about the well scanning mode on the plate readers for cell-based assays. Is this approach fast enough for viability screening?
Absolutely. Yeah, I mean, a lot of the times when you look at viability dyes, you only need to really report on a relative basis comparing one well to another. So yeah, you could be looking at certainly a 96 well plate in a matter of a few minutes. And even multiple colors, live dead assays and things like that could be captured very quickly. And these data files are very small. So there are sometimes people are using imaging approaches when there maybe isn't a requirement, for that level. You don't have to look within a cell to look at these things like viability markers. So it can be a very quick and fully quantitative way to look at a well very quickly without having to take lots of fields of view. Yeah, hopefully that answers the question.
Yes, thank you. And the next question is, how has the implementation of end-to-end automated processes improved data quality and reproducibility metrics, and how does this compare with a more traditional lab environment?
So we've seen on average a slight uplift in probably the more traditional QC type metrics like Z primes, robust standard deviations, and those sorts of measures, compared to sort of more traditional environments. Clearly eliminating the variability that we all carry in our in the way that we do things. I mean, serial dilution is probably the most variable process that exists. We all do it in a different way. We change tips, we don't change tips, we aspirate quickly, we cycle 10, we cycle 5. It depends what it is we're doing, but it's guaranteed that each of us probably does it slightly differently. And to standardize that is clearly going to deliver a level of an uplift in the quality and what we observe. I mean, we're starting to track more longitudinal performances. We've now been running sort of more in a commercial setting for about 18 months or so now. So we're building those data sets. We're hoping to share those shortly, but there's definite evidence that bringing that standardization in has significantly improved cellular, probably more than biochemical. But there are, the trends are all in a positive direction.
Thank you. We've got one more. I've got a couple more questions that have just come in actually, so I'll read those out to you. The first one is, could you please talk about the cost of test and drug development and the percentage of change with others?
So the first part I understand. I don't necessarily understand the percentage of change relative to others, but the cost largely is what I suppose ultimately quality costs. There's no reason to suggest that it should be astronomically higher, but equally, what we are trying to do is to improve decision making, and value is always in the eye of the beholder. So what one person sees as being an appropriate cost, another will think is too high, and that is something we're going to have to navigate. Clearly, we don't want, we see the service that we offer as being one that is premium, and as a consequence, we're starting to talk about more of a white glove service where we consider all avenues, all contributions of variability to any process, from collection of molecule, shipment, transit time across the world with a specialist logistics providers, ensuring that the molecule arrives and has traveled as we expect it to travel, whether that's a small molecule, whether that's a protein. These are all elements that are within the crosshairs of what we're trying to ensure, because data quality can look good and be underpinned by very poor reagent quality. So whilst it might look good, it's still meaningless. So the quality of data is only ever as good as everything that runs before, which is why the cost of the operation that we run does look to involve world-leading reagent providers, logistics providers, software, automation. We're not going to cut corners, because ultimately it will be to the detriment of the product that we sell, which is the data. And comparatively, I think that's a very subjective question. We don't have a huge number of answers there, but we're not 10 times more expensive. We're not a 10-fold cheaper. I mean, it's that is something that ultimately will come down to how much somebody values the project they're trying to run with us.
Yeah, I can imagine it would vary as well on the activities that you were looking to work through as well. So.
Absolutely. I mean, the more one requires FTE input, largely the more expensive a project's likely to be, because ultimately the biggest driver of cost is going to be infrastructure and personnel. But at the same time, that also brings with it the variance that leads to poorer decision making. So it's a balance.
Okay, thank you both for that. The next question is, it's clear that collaborative strategies in cloud labs will change the drug discovery process in quite a successful way, as we've seen, but it won't happen without overcoming resistance and reluctancies. What sort of resistance do you expect, and how do you think it might be overcome?
That's an excellent question. I think there is always, externalization or outsourcing, whatever you want to call it, has had a fairly checkered past, and that is largely because I think initially people saw it as a means of gaining access to flexible resource. A lot of it was driven out of markets and companies that were set in the Far East, and then came concerns over quality, time zone differences that meant that it was very difficult to speak or to vet processes and to ensure that what was being done was what was wanted. I think there is a much more of a globalization to it now. So whether we're talking CROs, whether we're talking service providers, there are as many of them across the globe now offering complementary and competing services. So clearly it's going to, because of current circumstance and the way that things have panned out, it's been given a boost. I hope it continues, and I think that the signs are incredibly positive that whether it's supporting smaller organizations, potentially a man and his computer, virtual companies, AI-led chemistry outfits. Clearly, in order to build the best models, you need the best data. There's no point in trying to train models off data sets that are flawed. So yeah, we have seen a sort of a renaissance and an acceptance that we're here to stay. There will always be those who still have their reservations, and I think there's always a conversation to be had whether people have reservations or not. I will never convince everybody that externalization is the way to go, but at the same time, what we're hearing in the marketplace is that there isn't a sort of a groundswell of naysayers that say the quality of data is not equivalent or better. It's going to take time, but I think the pandemic has kind of kick-started it, and we can see from companies that have started over the last sort of 2, 3, 4 years, as they hit seed raise stages, those raises are getting bigger. So clearly the investors see that as the future, and that's a very bright prospect for all of us who are in that arena looking to collaborate, and ultimately for patients at the end of it who will benefit from the quality of data we generate. So I do think it's here to stay for sure.
Okay, thank you. And the final question that we have is, where in the drug discovery process do you think there is room for the most improvement? Is it the very early stages?
Well, I think we often look at sort of molecules, and the more detailed work tends to happen later on. But if the first experiment we ever conduct is flawed, any work that flows from that is largely dead. So I would argue that the first experiments we ever do have to be the best controlled and the highest quality. So for me, the greatest improvements will come from the diligence around target selection, the diligence around validating those targets and getting some direct, be it genetic or disease association. But at the same time, quality should never slip. But there's absolutely no harm in bringing that quality to the front end and saying, if the first experiment I do is going to be of the greatest value, then tolerating a lower level quality index just because it's a high cost, high complexity experiment like a high throughput screen, for example, would seem to be a fundamentally flawed argument. So I think the greatest advances in terms of quality need to be made in the earliest stages, but not to forget that actually quality has to be central to every scientific discussion.
Okay, thank you for that. I'd just like to say thank you to you both for such great responses and such informative presentations, and also thank you to everybody who submitted questions. If you'd like to listen to this webinar again, it will be available on demand at ddw-online.com. You can now view Drug Discovery World content online on your mobile or tablet, or via the new DDW app, which can be downloaded via the App Store or Google Play. Thank you for listening.